My name is Alexandra Lucas. I'm a professor at the University of Florida and a practicing cardiologist there. At this stage, I've been doing a lot of research, and I presented it today at the International Academy of Cardiology, the 20th World Congress on Heart Disease in Vancouver, which is an excellent meeting. The work we've been doing has been going on for 25 years, and what I talked about was an overview. And the intent of the talk was to provide stimulus for discussion of pathways that are shared, that are involved in dysregulation of immune and inflammatory responses that drive not only heart disease and atherosclerosis, but also cancer and wound healing. And we have spent many years studying viral anti-inflammatory proteins called serpins, serine protease inhibitors. And we actually have taken one all the way through to a clinical trial and proven it was safe. And we've also used it in multiple animal models, and that is where this initiative in looking at overlap between these diseases, when you have dysregulation of immune responses and coagulation responses, they all interact. And so we talked about the ability to use a viral serine protease inhibitor, or serpin. It's taken from a virus. It's not used with the virus. It's a purified protein. And one of the proteins, this serpin, inhibits the clot-forming and clot-dissolving cascade. And we have a second one that inhibits the inflammasome and the cell death or apoptosis pathway. And we have data showing reduced damage after balloon and stent in atherosclerotic disease in a wide range of animals, rats, mice, pigs. We also have data after transplant with improved transplant survival, reduced scarring, reduced transplant vasculopathy with two of the proteins, two different serpins. We have cancer effects in a mouse model, human cancer cells with reduced growth with one serpin. And more recently, we found that we could improve survival with a lethal infection with another virus. So this one viral serpin from one virus can actually block the growth and improve the survival from a second lethal infection in mice. And finally, this same protein's been taken in through a small clinical trial in people, and it was safe and it reduced markers of heart damage. So we've got a wide range of efficacy, and I think what it shows us is that there are probably links and pathways that are shared in many of these disease processes, and that we may be able to follow the lead of the viruses and look at the pathways they point us to.